Okay, good afternoon.
Good afternoon, ma'am.
Okay. So for this afternoon, I will review you some important concepts in enzymes. Okay. Let's wait for a while. Some are still joining in. I'll give you 5 minutes. Please tell your class. Okay, so for this afternoon, for this morning, we will have enzymes. Okay, so a review. So if enzyme change active site, the rate of reaction will A, decrease, B, increase, C, remains the same, D, fluctuate. What is your answer? Anyone? Dawali, Mansi.
Doctor, increase.
If enzyme change the active site, the rate of reaction will increase, remains the same, fluctuate, decrease. So for you to be able to answer this question,
Doctor,
what you
decrease. A. Option A.
option A. Okay. What would you have to know for you to be able to answer this question? Okay. So you have to know what is an enzyme and you have to know the rate of reaction. Okay. In your study, so this is just a review. What would be the function of your enzymes? So these are things that you already know. I am just reviewing you. So this would be in a question and answer format type of review. Okay. So decrease. Okay. Why? What would be the function of your enzyme?
Ma'am, generally when enzyme attaches to its active site, the function will be enhanced as they have the similar receptors. Like we know already lock and key hypothesis of the enzyme, and the active site will have a particular shape which a correct enzyme will attach to it. So that's why whenever the particular enzyme changes its site to any different enzyme, so it may not attach properly, so that reaction will decrease.
Okay. So these are your active sites and your enzyme molecules, what happens? Contain a special pocket or cleft called the active sites. So if there is a change in this, so active sites, and it would not be a best fit for your substrate and your would be substrate complex, then it will decrease and will not facilitate your reaction. Okay? So if there's a change in your as we go back to the question, so it is asked in a way that if there's a change in active site, will rate of reaction increase or decrease? It will decrease. Now, if the question is if your enzyme enters a reaction, then what should your answer be? Then that is the answer of your other classmate. Your rate of reaction will then be increased because what is the definition of an enzyme? It is a catalyst of reaction and it tries to improve your rate of reaction by facilitating or making the reaction favorable or faster. Okay, next question. Okay, knowing that enzymes make reactions faster, so the general mechanism of an enzyme is that it reduces the energy of activation. B increases the energy of activation. C reduces your pH. D increases your pH. What is your answer?
Ma'am, it reduces the activation energy.
So it reduces the energy of activation. I'm pretty sure you remember the visual example of this wherein there's a high molehill. When there's an enzyme, it gets flattened so that you could push the boulder easier through that hill. And that is the visualization of reducing the rate of energy activation. So enzymes increase reaction by decreasing the activation energy. Thereby, the enzyme substrate interactions are formed. Remember also your lock and key model and your induced fit model. So your lock and key model, as I've mentioned in your active sites, those are your already ready. There's a corresponding lock and key. Induced fit as your enzyme approaches your substrate or your product enters your substrate, it makes your substrate and product complex possible by eventually altering its configuration so that it would be a perfect fit. So those are your two concepts of activation. Excuse me, some are still joining. Okay, so this is the picture. So enzymes lower a reaction's activation energy. So this is the mountain. So without your enzyme, your lactose, for it to be glucose and galactose would have to have a certain activation energy for you to be able to produce the reaction. Okay. Okay. With your enzyme, lactase, so your lactose forms a substrate enzyme complex and releases your glucose and galactose, thereby lowering the activation energy, thereby making the reaction easier. So that is how we remember your activation energy. So it is reduced when undergoing your enzyme reactions. Okay. This is also another visualization of your reduction of activation energy. So there's the without enzyme and with enzyme. So remember, delta G is unaffected by the enzyme. Okay. Okay. So this one, the coenzyme is often a metal, always a protein, often a vitamin, and often an inorganic compound. Okay. So you have to remember that your enzyme does not work alone. So it has different cofactors. One of each would be coenzyme as an example in this question. So how do you answer this question? Anyone who wants a guess?
Ma'am, it is an inorganic compound. It is not always a metal, not always a protein, and not always a vitamin. So it may be an inorganic, maybe sometimes protein, maybe sometimes vitamin also.
So you
So
are
option is D. Yeah, yeah.
asking
Option is D, ma'am. Often an inorganic
in
compound.
organic compound. Other answers and other ways that you arrive at the answer?
Doc, often a
Often a B, someone
vitamin.
answered B, always a protein. So you're divided. Often a vitamin looks like it's the first time you encountered this analysis question. Okay. So these are your types of cofactors. Your coenzyme, non-protein component, loosely bound to your apoenzyme by a non-covalent bond. Examples are which are a vitamin or compound derived from vitamins. Okay. The other is a prosthetic group, non-protein component, tightly bound to your apoenzyme by covalent bonds, and it's called a prosthetic group. So going back to our question, so this answers a vitamin. So your coenzyme is a non-protein. So that by deduction, you could answer. Okay. So an enzyme that joins the ends of two strands of nucleic acids is polymerase, ligase, synthetase, helicase. So for this, for you to be able to answer this question, what do you have to know? You have to know the structure and the function of your different enzymes. And what would this enzyme that is described do? It joins the ends of two strands of nucleic acids. What's your answer?
Ma'am, ligase.
Ligase, ma'am.
Yes, ma'am.
Ligase. Okay, this is something looks like a lot of you would know the answer. So what's the answer? Ligase. Oops. So another function, enzyme function question. So enzyme which helps in changing shape of molecules is called
Isometrics.
Isomerization.
changing shape, isomerase. Okay. So for you to be able to answer this question, you have to know how it is classified. Okay. Some would do the number, but for your benefit, I think it's beneficial that you know the names because more or less you would know the function just by knowing the names. So the functions of different enzymes would be your oxidoreductase, your transferase, hydrolase, lyase, isomerase, and ligase. So the first question, what's the if you do not know your um, if you do not know your enzyme functions, you could be led to answer synthetase. Two strands of nucleic acids, you will join it. Oh, I will answer synthetase, you will synthesize, etc. That's if you do not know the list of functions of enzymes. But if you know the list, you will not be confused with that, and indeed the answer is like this. Okay, of course you should know the function. Any change in shape, that's your isomerase. All right, so those are your function type questions. Next, so which of the following is produced with the combination of an apoenzyme and a coenzyme? Holoenzyme.
Holoenzyme.
So you're sure with the answer, holoenzyme, substrate complex, prosthetic group, enzyme product complex. Okay. So you know this, looks like you know it at the back of your head. So apoenzyme and holoenzyme would be the enzyme without its non-protein moiety and is termed as your apoenzyme, and it is inactive. So holoenzyme is your active enzyme with its non-protein component. Okay. So again, types of cofactors. Your prosthetic group is explained. That is what your prosthetic group is. Okay. Again, remember your competitive inhibition and your non-competitive inhibition. This is very please don't draw on my screen. I think it's CD. Okay. So competitive and non-competitive inhibition. So you have, like it or not, you have to be very familiar with different reactions. And the only way that you go by it is be familiar with your different cycles. Um, Always remember the rate limiting steps so that you know which is competitive or non-competitive, because these are the usual important reactions in your um in your cycles. Okay, so what's the answer?
Option A, succinic dehydrogenase by maluric
Succinic
acid.
dehydrogenase by maluric acid
Yes,
is
doctor.
competitive. Does everyone agree?
Yes, ma'am.
Non-competitive. Okay. You know your G6P, your glycolysis. Okay. So This is your reversible competitive inhibition. So your competitive inhibitor has a structure like the substrate. So it enters, competes with the substrate for the active site, and has its effect reversed by increasing your substrate concentration. Okay, so remember the properties of your enzyme. So remember your Vmax and Km in relation to your competitive and your non-competitive inhibition. Okay. My analogy for this would be, for example, a relationship. If one is um, more associated with the other, for example, this substrate and enzyme complex, you could liken it to a relationship, girlfriend or boyfriend. Okay, you want to get in the picture. So what do? You increase your efforts so that this substrate complex would break off and then you enter. So you could reverse it by competitive inhibition by increasing efforts. On the other hand, non-reversible component, it has a different configuration. Do I have of that? Okay. It has a different configuration. So even if you have your substrate available, but you cannot reverse it because it has a different configuration. So even if you increase your efforts, if it is not suitable for you, you cannot change the reaction because of its different structure. So a non-competitive inhibitors has this following qualities. Okay, so next. While bound to the active site, substrate is converted into the product of reaction, complex, another substrate of high energy, or all of the above.
Product of reaction.
Again, I'm sorry. It's the
Product of reaction, ma'am.
product of reaction. Okay. So remember your enzyme substrate complex. Oops, I'm sorry. It has a step one, enzyme and substrate combine to form a complex. So this is your enzyme, this is your substrate, and you have a complex. Okay, then it forms into your enzyme substrate complex, and then it produces your reaction. Okay, and your product would be the result of the process. And again, your enzyme, as it follows your Michaelis-Menten kinetics, it is ready for another substrate and it is not consumed. Okay. So enzymes do not work in high temperature because enzymes get denatured, the active site changes conformation, the substrate gets inactivated, Vmax has been reached.
Option
So
A,
what
the enzyme can be denatured.
you have to know your enzymes and the factors that affect enzyme efficiency. Okay, so these are what affects your enzyme activity. There are three factors: environment, cofactors and coenzymes, and enzyme inhibitors. So, what's your environment conditions? So, extreme temperature, you know that it's protein. So, at extreme temperatures, particularly at 40 degrees, your protein or your enzyme gets denatured. And that's the answer to the question. Okay? So pH. So most like your 6 to 8 pH, which is near neutral, except for some. What are the exceptions that you will know of? What are the exceptions? For example, what are your stomach enzymes that would prefer
Pepsin, Doctor, Pepsin.
acidic? So your pepsin, on the other side of the scale, your trypsin would like alkaline or acidic environments. And your substrate concentration. And this is where your Vmax will enter. So there comes a point when you add your substrate, as we have learned, reaction gets faster. But why is there a Vmax, or why is there a limit in your rate of reaction as you put on more substrate or as you add on more substrate? What concept is that? Okay, have you ever come across the concept of supersaturation? So that explains your Vmax or your maximum concentration, wherein anything that you add more won't affect the rate of reaction because your receptors have all been used up. It's the same way that your in physiology there are refractory actions because your receptors have been also used up. Okay, so this is the optimal temperature. Anything more than your 37, 40 would denature your proteins. Okay, this is your pH. So your pepsin and trypsin would want acidic or your alkaline environments. And this is your maximum velocity. So enzyme is saturated, far more substrate than it can deal with, it will not change your reaction anymore because all of your receptors is already combined with your substrate. Okay. Cofactors and coenzymes. So these are inorganic substances which facilitate your enzymes to work. So these are, as mentioned, some vitamins, etc. So example, iron must be present in the quaternary structure hemoglobin in order for it to pick up oxygen. Okay, so that is your review on coenzymes. Will you have other reviews this afternoon?
இன்னும் கொஞ்சம் வெளில போனா நல்லா இருக்கும். டக்குன்னு வெளில போ.
Okay.
இல்லப்பா வெளில போ.
Okay. Okay. So I would just like to emphasize some concepts of enzyme kinetics as a review. Okay. So you know what enzymes are? Protein catalyzing reactions without changing the overall process. Okay. My favorite analogy of this would be a community development worker. Okay. You go into a community, facilitate change, but in the end you have to leave the community because your community would be would continue to exist without the need of your presence. So that is an enzyme. So like it or not, your enzyme kinetics is mathematical quantitative measurement of your rates of enzyme catalyzed reactions and the study of which. Okay. Okay. So these are just terms. We have discussed that. This one also. Okay. I want you to concentrate on your Michaelis-Menten equation. Okay. Michaelis-Menten has three assumptions. I'm sure in your biochem books you have read it and it's very wordy. And I want to summarize it and how you understand it in these simple concepts. Okay. Anyone who wants to try to guess what this is in the assumption of Michaelis-Menten equation? S
Ma'am,
is for
concentration of
substrate
substrate.
and your enzyme. So, what is the relation of the concentration of your substrate to your concentration of enzyme by this visual representation? So that you don't have to memorize the whole gamut of your assumptions. You just have to understand this. Okay, you say the concentration of your substrate, continue. Continue with your line of thought, the one who was answering, or anyone who would want to fathom a guess.
Ma'am, it's showing the relationship between the substrate concentration and reaction rate.
Sure. This very big S is your substrate, and this is your E. So what happens if you have a very big number compared to a small? And this
Indirectly
leads,
proportional to it.
and this leads to your first Michaelis-Menten equation. The substrate is so big that your substrate concentration of your enzyme concentration becomes negligible in the equation. Thereby, when you compute it, it's just your substrate. Okay? So your enzyme substrate, so this is your second assumption. It does not change with time and rate of synthesis is equal to rate of degradation. Okay? So remember that. And the third one, initial reaction velocities are used. And if your initial reaction velocities are used, it is such that your end equation is almost zero because this is where the velocities are at its peak. So initial reaction velocities are used, thereby decreasing that side of the equation, thereby you have almost a zero. Don't worry, we won't ask you to derive your Michaelis-Menten equation. So your Km is your Michaelis constant, and this is characteristic of an enzyme and its particular substrate. So this reflects the affinity of the enzyme for that certain substrate. So remember, your Km, small km, large would be low affinity. Always remember that. If it's L, low affinity. If it's a small, then this is have a high affinity. Okay. So it's good if your Michael-Mentel's equation would be for single substrate. However, you know that in biochemical reactions, it is always multi-substrate. Okay, so always bear that in mind. Okay, so this would be low concentration. So if it's low concentration, the velocity of the reaction is proportional. So that means it's proportional to your substrate concentration. But when it reaches your Vmax, it is zero order. That is the constant and independent of your and your Km is independent of your substrate concentration. Okay, so these are nice to know. Okay, I would like to bring you to your inhibitors. Okay, maximal velocity, so that is your Vmax, is decreased in the presence of a non-competitive inhibitor. Km is unchanged. So this is in non-competitive. Non-competitive, Km is unchanged, Vmax is decreased. As to your competitive, Vmax is the same while Km is increased. So non-competitive, Km is what? Unchanged. Your Vmax is decreased. Okay. Remember those four relationships so that when you encounter it in questions of competitive and non-competitive inhibitors in enzymes, you do remember. Okay? I will send my lectures in Biochem so that you could review. I will send the links so that you could review some lectures of biochemistry for your seminar. Okay? Any questions? Any clarifications? I hope I simplified some concepts for you this morning. If you do not have any questions, you may leave this call. Thank you for attending this review and good luck with your exams. Okay? I will ask your, sorry, who's the Biochem LO, but I would ask Prabhav and Sai to disseminate the links. I will send it to them. I'm sorry that those are the two people that I have access to. All right? Any questions?
No,
You may leave this call. Thank you.